Hello everyone, greetings from the Dinosaur Soft Tissue Research Institute. We are very excited to announce groundbreaking new work that is going to be featured, particularly in this video, and hopefully to be published soon. It features the ongoing work of JimSolliday. Jim is a personal friend, known him for about 30 years, a fellow microscopist. He's the president of the Microscopist Society of Southern California. He's also the winner of the prestigious McCrone Research Associates Award for microscopy. And DSTRI is very excited and pleased to announce that JimSolliday has joined the Dinosaur Soft Tissue Research Institute as a senior microscopist. He's been working for the last 3 years on not only the Triceratops specimens that were produced in my work, but also he's collected very old bones, some 160 million years old, if you adhere to the time scale. And his results are rather stunning. And so without further discussion, I present to you JimSolliday, Senior Microscopist at the Dinosaur Soft Tissue Research Institute.
Is that the complaint is that you creationists never do your own research, you just criticize other people's research. So you guys know that's not true. We actually do a lot of research. Tonight you're going to see original research. The part that I did is not yet published. It's going to be. The part that Mark Armitage did has been published. This project initially was known as the Dinosaur Soft Tissue Project, and it was started by Mark Armitage about 4 years ago. Of course, I've known Mark Armitage for about 30 years. He's also a member of the Southern California Microscopist Society. And we had a conversation one day, and he introduced me to the Dinosaur Soft Tissue Project. And I was skeptical. I asked if he would let me review his material. And so he provided me some of his material that he obtained. Now, the way he obtained material was using Dr. Mary Schweitzer's technique of taking a fossil bone sample, putting it in a petri dish, and dissolving it in a weak acid. When the bone matrix is dissolved, it leaves a suspension of soft tissue or proteins. Proteins don't dissolve in this kind of acid. So we made slides. I looked at the material. I spent about 2 weeks studying it and was completely convinced that it was legitimate. I got involved in the project. We've been collaborating ever since. And to summarize the project, the first phase is actually going to be in 2 phases. The first phase was to use Dr. Mary Schweitzer's technique of dissolving bone, extracting the soft tissue, and seeing what we have, basically proving that soft tissue still exists in dinosaur bone. The second phase, which I did, was finding soft tissue in situation. And that's why it's taken about 3 years. So most of that time was used up obtaining material and getting thin sections made. So what we did was we would take a dinosaur bone, we would have a thin section, and we would do it in a very specific way that would allow the soft tissue to stay. Mostly when thin sections are made, there's a protocol that was established by Ellen Lamb. She's the histologist at the Museum of the Rockies. And in that particular protocol, bone is cut into as thin as possible section, it's fixed, and then it's embedded with the plastic to keep it firm. So when you grind it down to a real thin section, it doesn't fall apart. Well, the problem is when you embed this bone, you have to then dissolve it out, to mount the slide. So in dissolving it out, that carries all the soft tissue with it. So we had to come up with a new method, and that's what you're going to see tonight. And so we did manage to find soft tissue in thin sections. That's phase 2, and that's what you're going to see tonight. Be up to date on that. Phase 3 is the immunofluorescence part of the project, and that is based on an antigen-antibody reaction, where you can take a stain that's really an antibody that will attach only to one thing, and that's specifically to a protein. So if you're interested in verifying that there's type 1 collagen, You have a stain that's an antibody that will attach to the receptor sites on that particular protein, and then when you use ultraviolet radiation, it will light up, so you know for sure that you have that protein. So that's what we're doing. That's the phase we're in right now. So I will get started, and we'll bring you up to date on the project. Okay, to begin with, I started out with this picture here. This is a stegosaur, and it was carved into a column in a temple in Cambodia. And the interesting thing is, it's made of stone. Now, when I was in college, I got the impression there was no such thing as dinosaur bones. And the reason is that we were taught that all dinosaur bones have been subject to a process called diagenesis. In that process, all the bone mineral and all the proteins and all the collagen has been replaced by dissolved minerals. And so what you have left is a cast of the bone of minerals that infilled and basically replaced the bone. So basically I understood there was no such thing as a real dinosaur bone. It was all fossil. Well, turns out that's not quite true. Okay, this is what we're going to talk about tonight. Introduction, do a review to give you some of the terminology I'll be using, so you have you'll be able to place that terminology with the picture. Second, we'll look at actual bone cells extracted from dinosaur bone. Third, we'll look at bone sections to look for soft tissue in situation. And then we'll do some perspective. We'll try to figure out what it means and what it took for a dinosaur bone cell to survive for 65, 70 million years. Okay, a little bit of a review on bone material. This is a femur bone, and there's a little piece taken out here, and it's enlarged here. Cortical bone on the outside, soft bone or spongy bone on the inside. It's actually a clavicular bone. You have the bone matrix itself. The cortical bone is made up of what's called Haversian tissue, and that's made up of these oxyons, these little round concentric lamella that have a canal down the middle called the Haversian canal. And the blood, nerves, and oxygen come through this canal and feed all the little bone cells that are inside what are called lacuna. These are little chambers inside the lamella, inside the oxyon. And these little bone cells keep the bone healthy and monitor the bone. This is what it looks like under the microscope. One of these osteons, here's one here. The canal, that's what it looks like right here. So you'll be hearing these terms as we go. To make things even simpler, this is cortical bone and this is spongy bone. You'll be seeing a lot of that type of bone. You'll be able to recognize it and remember. The inside surface of the bone is called the endosteum or the endosteal surface, and that's where I found a lot of soft tissue, so try to remember that name. The outside is the periosteum, and here we have the Haversian system or the osteons and the canals. Here's a little wedge of what one of these osteons looks like a little closer. You have the lamella, these different rows here, and the osteocytes are in the lacuna. This is a close-up of an osteocyte. This is important to remember. This is an isolated osteon, and each lamella, the collagen fibers will run 45 degrees opposite each other. This gives the bone a lot of tensile strength. So you'll see the collagen will go to the right on this lamella and it'll go to the left on that lamella. That will become very important later on. This is what a living bone cell osteocyte looks like alive, or recently alive. Here's a bright field section and molecular micrograph section of osteocytes with their filopodia. These are the 4 living bone cells. A little quick review. The osteocytes are in the bone matrix itself. The osteoblasts are the cells that actually deposit the mineral. They actually paint themselves into a corner, and when they've deposited bone all around them, they turn from an osteoblast into an osteocyte. So here they create bone, and once they're trapped, they end up monitoring and keeping the bone healthy. This is an osteoclast. That's part of remodeling. These are the cells that actually dissolve the calcite and recirculate. calcium. Okay, this began about 5-6 years ago when Mark Armitage was able to recover bones from a triceratops in Hell Creek, Montana. A couple of important notes here. Right now they're digging up a triceratops horn, and I think the Lord had a hand in this because it's extremely difficult if you find a fossil bone out in the wilderness in Montana. There's almost no way, unless you're very experienced and you're able to spend a whole 6-7 months there extracting all the bones and put it together, it's almost impossible to tell what genus of dinosaur you have. Well, the Lord gave us a triceratops with a horn that's very obvious, so now we know exactly what genus of dinosaur we had just by finding one single bone, and that's almost never done. So the bone itself is around 2 feet under the soil. Part of the horn was sticking out. There's a little ravine off to the side here. It's probably about 6 or 7 meters down into the bottom of that ravine. And on the side of this little ravine, the triceratops horn is actually sticking out. And a woman named Marjorie, who owns this ranch, that's what she actually saw, and that's when she contacted us and said, you might want to come and extract this bone. So that's where the material came from, the original material came from, that gave us the original soft tissue in our particular research. Okay, a couple of things you need to know about fossil bone. There's basically 2 types of dinosaur bones that you can work with. This is called agatized dinosaur bone, and as minerals are dissolved and they're deposited inside the bone, that's a process called diagenesis. Diagenesis occurs primarily by silicates, quartz, chalcedony. It's very hard, and you can't really digest that with an acid. Here's a couple of examples of agatized bone. Most of that is found in Utah, most of those bones. Very beautiful bones. What we're interested in is the type of diagenesis, or the type of fossil that is primarily by dissolved calcite, carbonates, and other salts. These are found in the Hell Creek Formation, Montana, and Dakotas. You can see it's not a pretty bone, but this is where you can find soft tissue. So you have calcite deposited in this one, and silicates in this one. This is not good. This is the kind we want. So that gives you an idea of the type of dinosaur bone we need. Okay, optical issues and techniques. Dinosaur bones turn out to be one of the hardest specimens to acquire high-resolution images, high-resolution microscopy. There's a lot of reasons for that, and I'll summarize. First is the slides. I've got a slide over here on the exhibition table. I actually have this slide on the table, as you can see it. It's about 5 inches by 4 inches. It's an extremely large slide, so you have to have a special stage in your microscope. That's the first difficulty. The next difficulty is it comes in all different types of slides. This is the ideal slide with the cover slip. Microscopes' objectives are designed to be used through a cover slip. As a matter of fact, the optical equation that's used includes the cover slip as part of its opticals. So to get a good sharp image, you really need to have a cover slip. Unfortunately, these large slides and dinosaur bones very often are too big for cover slips, you know. So you have to find a way to take images without a cover slip. So that means special objectives. The other thing is the sections can be extremely thick. Microscope objective will only focus on about maybe 7 or 8 microns, very, very tiny depth of field. So it's very difficult to get good images when the specimen has to be so thick. Okay, uncovered specimens need to be studied on both the exposed surface and from the bottom through the slide. If not, you'll miss a lot of good information. To resolve this condition, long working distance objectives with correction colors are required. These are special objectives. They're about 3 times more expensive than a regular objective, and they're actually designed to look through an entire microscope slide. That's why they're so expensive. So you have to start out by getting special optics. You can also get optics that are designed to be used without cover slip, and actually the best ones for that are what's called epi objectives. Epi objectives are made to look at metals, if you're a metallurgist, but they work very good for uncovered dinosaur bones. Again, uncovered slides, and then For a covered slide, you want to use a regular polarizing objective. This is an objective that's designed to be used with a cover slip. And the problem with it is you only got about 8 microns below the cover slip that you can focus because the depth of field is so small. So you end up having to use the more expensive objectives. So, but probably one of the biggest issues is called spherical aberration. When you use a slide that doesn't have a cover slip, you introduce this aberration. If you use a cover slip, you don't have the aberration. Here we have a picture that was taken with a regular objective on a slide that did not have a cover slip. What this is a canal, one of those Eversian canals. This is bone matrix here. These are lacuna, where the bone cells are. And this is fungal hypha that's penetrated and infected the bone. You can see everything you need to see, but it's not really sharp. Now, if you take this with an objective that was designed to be used without a cover slip, you can see it sharpens up. You just eliminated that spherical aberration. Here are the two pictures side by side. You can see the detail here compared to here. This is a little fuzzy. That fuzziness is spherical aberration. So it's a big problem. Every single slide you study requires a whole different physical setup with the microscope. The final problem is ground fossil bone with cavities that are filled with calcite. Okay, when fossil bone has not been decalcified, which is desirable, the cavities remain filled with calcite, which has a dual refractive index. Aberrations represented as double imaging can occur, especially with high magnification. So you see these little framboids, they're little iron pyrite framboids in this canal. And this, by the way, is an osteon, and here's one of the lamella, and here's one of the central canals. So we go to high magnification, you can start to see these double images. See the little green here, see it's red up here and blue down here? That's because calcite has a dual refractive index. It gives you problems. So you can eliminate that by using a dichroic filter, but then you don't have any color. Okay, I wanted to start by showing you the worst fossil bone that I have. This is from Morocco, and it's the most lithified and degraded bone that I have. It's Cretaceous and comes from northern Africa. However, the histology is still amazing. You still have the central canal, you still have the concentric lamella, you still see the lacuna where the little bone cells are, you even have a cement ring around here, and interstitial lamella out here, so you still have all the morphology. This is a lower power, but you can see that a lot of the bone is literally dissolving away. This happens when the bone is exposed to moisture, so every year it rains, water goes through the sediments where the fossil has been and starts to leach out the bone. So eventually the fossil bone will completely disappear. In this case, all of the hydroxyapatite has been replaced by calcium carbonate. This is a very important picture because this sets the standard for the research. This is my control slide. This is the rib of a dolphin, and it's a recent bone. The dolphin was found on the Isle of Wight in the UK. It had been dead about probably a week to 2 weeks, and most of the flesh had been rotted away through the process of taphonomy, which is the description of how things rot. A rib was exposed and was collected, and a thin section was made. Now the reason this is important is this is recent. This is something that's had 2 weeks of taphonomy or 2 weeks of decay, and so I want to see what the best situation I can expect would be. What kind of soft tissue is left after only 2 weeks of decay. Here you have an oxyon and the concentric lamella. You have the Eversian canal. You have all these osteocytes and their filopodia. These filopodia actually are little tiny tubes called canaliculi. They're end tubes that go to these canals and they collect nutrients from the canal, and then they feed themselves. Then they pass what they don't use on to the outer cells. This keeps the bone cells alive. Looking at this sample, oh, by the way, no processing was done. This bone was cut into as thin as possible, one side polished, cemented to a slide. A little bit of formaldehyde was used to fix it, just so it wouldn't stink. The next thing is it was ground down to about 80 microns thick. Then it was washed with a little bit of alcohol, and a little bit of xylene was added, and some Canadian balsam and a cover slip, and that's it. So there's no decalcification and there's no embedding. So you're getting an unspoiled sample. So I started looking around what soft tissue is left here. The reason the Philopodia and the osteocytes are dark brown is because as soon as the animal dies, the proteins start to oxidize, and it's just like rust, they start to turn dark colors. I'm seeing a little bit of material here, I'm seeing some stuff here, I'm seeing some little bit of material here. This looks like the best one, so notice you have a osteocyte here and two little osteocytes here. So let's look at this one a little closer. Here's that two, and here's the two over here. I'm trying to determine what's in here, what soft tissue is left. We know that the osteocytes are still embedded in the lacuna. Let's look a little closer, and we have red blood cells. So I know now, the question was, would red blood cells remain in these canals? The reason that's a question is because when an animal dies, goes to the ground, gravity begins to work immediately. And as long as the blood is fluid, gravity starts pulling all that blood down to the lowest center point. So you have what's called post-mortem morbidity. So the blood technically should all drain out. It takes about 20 minutes for the natural process of clotting to start. Once the blood starts to clot, then it can probably stay where it's at, cement in place. So I know now that I can expect, if I'm lucky to find red blood cells, are going to remain in the canals. Now you can see them really well. Here are where the Philopodia connect. There's little valves, little micro valves here, and they take albumin and other proteins from the plasma and feed, and they send it on to the other cells. One thing that happens here, when the animal dies, all the blood vessels they relax and they expand, and they end up submitting themselves to the inside of, or the endosteal wall of the cavity. In this case, a canal. So there's a little bit of a blood vessel left around the edge there, and then there's blood cells here, and then the cavity wall on the outside. Now on the cavity wall itself, it has what I call endothelial cells, and they are made of protein, so that's going to be of interest to us down the road. This is a different type of illumination. This is phase contrast. Okay, a little bit of review. Dinosaur soft tissue, discovery of dinosaur soft tissue background. In the 60s, people started seeing what they thought might be soft tissue. Mary Schweitzer published in the 90s suggesting original proteins still existed in dinosaur bones. But by 2005, Dr. Mary Schweitzer of North Carolina State University published her discovery of exquisitely preserved soft tissue in dinosaur bones. She found that much of the tissue even remained elastic. She also found intact blood vessels, also with individual dinosaur cells, bone cells, osteocytes. She also preserved or intact original dinosaur proteins, as well as evidence of fragments of DNA. This is later on. If you go to Wikipedia, Wikipedia states that the extraction of protein, soft tissue, remnant cells, and organella-like structures from dinosaur fossils has been confirmed. Now the reason that's important is because I found 5 or 6 articles online recently that say no one's been able to reproduce Dr. Schweitzer's work. Well, that's completely false, and you're going to actually see her work reproduced here tonight. In the past, we were taught to think of a fossil as having original bone material replaced by minerals. The presence of actual bone, let alone pliable blood vessels and blood cells and protein, is unthinkable. So what did Dr. Schweitzer actually find? pliable, flexible, soft tissue, collagen. Her video actually shows tweezers. She spreads that apart and it comes back and forth. It's still flexible. This is from a 67 million year old T-Rex. Transparent branches, hollow blood vessels corresponding to vessels found in modern animals. Original dinosaur osteocytes or bone cells. And finally, she found hemoglobin in red blood cells. So, can that be duplicated? These are ostrich and emu soft tissues after bone has been dissolved in acids. This is the material you can see. These are the soft tissues extracted from her T-Rex after acid treatment. And you can see these blood vessels, the T-Rex blood vessels, millions of years look almost better than the extant blood vessels. After dissolving the mineral portion of the bone with weak acid, various types of flexible structures were recovered. They conform to microscopic pores of the bone from which they were liberated. Okay, acids used. Ethylene diamine tetracetic acid, EDTA. It's actually it does two things. It's a weak acid. It's also a chelating agent, so it acts as a fixative, and it captures calcium ions from the surface of the apatite crystal. That is the material we use. You can also use hydrochloric acid, which actually works a little faster. You can use 10% hydrochloric acid, which is the cheapest way to go. And then to stop the digestion, you can use ammonium bicarbonate or sodium hydroxide. Digestion time is determined by monitoring, so you have it in a Petri dish and you put it under a stereoscope and you look and check and see how much is gone. Our particular work took between 4 weeks and 4 months. Suspended products of decalcification were washed and then mounted for light microscopy. Here's what dissolved bone, as it's dissolving, looks like, as it's disappearing. Here is a bone chip with an osteocyte still in it, not quite liberated yet, but you can see that there's a beautiful osteocyte still inside that bone. This is from the Triceratops bone that we covered from Montana. Here you can see the etching. You can see a bone chip being dissolved from the surface out. This material here is actually fibrous collagen that's been liberated from bone. These are very microscopic bone bits that are left. This is taken under the microscope. Okay, cell diagenesis and preservation. I want to talk a little bit about these bone cells and what happens to them as they fossilize. And just to give you an idea of the material that we were working with, this is Bob. He's a museum specimen. Materials that are parts of this anatomy that were recovered was a 42-inch Triceratops horn, part of the frill, rib bone, and there's a piece of rib bone up here to look at at the exhibition table when we get done. And the condyle was recovered. The condyle is a round bone that the head is mounted on, that rotates on. It's very, very rare, very valuable. Mark only had 3 days, so that's all the material that was recovered. It's plenty to demonstrate the project. This is from a modern bone, and this is the ideal, perfect situation where the acid has dissolved away the bone matrix and left just the osteocytes and the filopodia. You can see how all these osteocytes, bone cells, are talking to each other through these little canals. Well, actual filopodia, the canal is gone. This is from our Triceratops. You can see that the osteocytes are still in excellent condition and filopodia are still present. This is fibrous collagen. They're actually sitting in fibrous collagen. This was taken with an electron microscope. Filopodia, which are these little things, they shrink and contract after death. Cameliculi, or the little tubes that they're in, are quickly sealed by dehydrated serum proteins like albumin and glycoproteins, and they end up turning dark brown, sometimes black. Okay, this is what you should expect. I'm going to show you everything we found. In time, bone degrades and allows water access to the lacuna. That's the little chamber that the bone cell is in. This invites dissolved minerals to quickly infiltrate space and calcify the cell. So water, through a process called hydrolysis, basically putrefies and gets rid of the live cell. And then calcite usually comes in and leaves this first order mineral. You can tell calcite from its color and its brightness. So this is what you should expect. This is another example, but it's in better condition. This is in situation, in actually a bone section. This has not been dissolved in acids. The high birefringence indicates a first order mineral, calcite. That's that bright yellow. The canalicleus, you see these little tubes around it, they're congested with oxidized and chelated proteins. They're turning dark. And then in the middle are calcified organella, distributed throughout the cell. Okay, these organella, these little tubes are only 0.2 of a micron thick in real life. The organella are way too big. They can't get through them. So the only thing is that moisture can get in, dissolve the cell, and then end up calcifying all the organella. So you have a nucleus and all the little cell parts are still there. Amazing fossilization. This is an extraordinary picture, by the way, for that reason. It's very rare. I've looked at over 100 dinosaur slides. I've only seen 2 of these beautifully preserved cells like this. This is the other one. Now this is a very important slide because it gives a lot of information that will help you understand what we're looking at. This is using polarized light. This is a thin section. The section is exactly the same thickness throughout. The difficulty is that the density of the bone isn't the same. Right here, you have a canal. It's hard to see. You can tell because there's calcified blood cells here. So we know there's a canal, and we know that it's yellow on each side of the canal. The yellow means that the density is very low, and that water has come through this canal every year, every winter, as it was in the ground, and it would damage this bone. Okay, this is a lacuna here. It used to have an osteocyte in it, but when the water got to it, the osteocyte perished and left. And actually, let me go to the next slide. This shows moisture has infiltrated the lacuna, decomposing the cell and leaving a fluid inclusion with a bubble of gas called methane. Methane is produced for putrefaction. And that's because it's been exposed to a lot of water. It's close to that canal. Here you have a transition zone of degraded bone to well-preserved bone. This yellow bone is less dense than this beautifully dark bone up here. And you can see all these osteocytes are gone, but this cell up here is still very good. That hasn't been exposed to as much moisture. Many of the osteocytes found in the well-preserved zones are mummified. They're not gone. After the surrounding bone is digested in acid, the elastic cells are released and isolated for study. So that's what we're going to look at now. This is the product of dissolved bone. We used Mark Armitage used Mary Schweitzer's method. This first picture is of a bone chip, because you take samples as you're going up. You don't wait for it to be completely digested. This is a bone chip with fibrous collagen still on it. It's obvious what that is. Shouldn't be there. It's protein. This is a fantastic find. This is an osteocyte still in a bone chip, and you can see how healthy these filopodia are. You remember that picture I showed you of the modern bone cell with all the filopodia, how many there were? All still beautifully present. It's still locked in the bone matrix, but you can see the osteocyte and the filopodia beautifully. This is a fully liberated bone cell, an osteocyte. You can see the filopodia, you can see the cytoplasm, you can see dissolved bone. If that wasn't there and we dissolved the bone, there should have been nothing there. This is one of the best pictures of an osteocyte of a 70 million year old fossil bone cell. You can still see the granulated cytoplasm. You can still see all the filopodia, a little bit of bone matrix left. This is in almost perfect condition. Bone cells come in three types. So when you're looking at a bone under a microscope, you can expect to see three different types of bone cells: type A, B, and C. Here's an example of type A. They're long and slender. And you can see fibrous collagen all left in the matrix. This is type B. They're a little fatter, a little shorter. And this is type C. They're almost round. So we found all three types of bone cells in our results. This is a type C, high resolution, high magnification. You can see the filopodia are still connected. You can see the membrane of the cell is intact, and you can see membrane is actually tightly fit around some of these organella still in the cell. When you press down on the cover slip, these little filopodia move, so it's still flexible. This is type B. Again, the membrane is intact. Filopodia are intact. There's a little bit of collagen here. This is another type B. This is even more interesting. Here you have one of the filopodia folded up over itself. Here you have a nucleus and a nucleolus still present. I want to mention about these little beads. Proteins, especially collagen, are fibrous proteins, and when you perturb the protein in osmotic pressure that's not used to, the protein will curl up into a little ball, like a sow bug. If you bother it, it will curl up into a little ball. Well, what proteins do when they're isolated from their matrix, they'll curl up in these little balls. So that helps verify that you're looking at collagen. You'll see these little balls all over the place, because once the protein's liberated, it's got the matrix and it starts falling apart, it will fold up like that. Here's a type B osteocyte. Now what's interesting about this, you can see here a little vacuole, and right there's another one, and the reason I mention it is because when I press down with the needle on the cover slip, this little vacuole here will open and close. You would never expect to see that in a fossil bone cell. Well, you wouldn't even expect to see the cell, let alone the fact that it's still flexible. You can see organella in the cell still. This one has got beautifully granulated cytoplasm, proteins all balling up like I mentioned, and there's a calcified nucleus in the middle. In all the literature I've read, 100 papers, and I've never yet found images that illustrate the cells quite like this. Here's a type A. All the filopodia intact. You can see some of that collagen over here. And you can see granulated cytoplasm. There's material still inside. It's probably calcified, most of it, but it's still there. I have over 100 images of, quote, fossil bone cells. This was using an electron microscope taken by Mark Armitage, an osteocyte from the Triceratops. You can see one of the filopodia folded on itself, and you can see another one under here. The matrix is fibrous collagen, and underneath that is bone. This is a super high resolution picture. You can see the detail. This cell is, you can look at a monitor and you'd be hard pressed to tell the difference. Now we're getting even more interesting. This is more of our liberated soft tissue. These are blood vessels. When I first looked at this, I thought, oh no, I've got a lot of fungal mycelia in my sample. And then I realized that, wait a minute, this has been in acid for about 7 weeks. There's no way that mycelia would be left. Also, Also, the shape is typical. When you look at the canals, if you've studied the canals of bones, you realize that they get smaller and wider. They have little knobs, all kinds of little structures. So when you look at this stuff closer, oh, this is actually Dr. Schweitzer. This is what the blood cells look like after they've been isolated from bone and they're floating in a Petri dish. This is taken with a low-power stereoscope. But you can see these little knobs. You can see these little. Here's a couple of isolated. See the little knobs. Now, what made me give up the idea of fungal mycelia is that soil mycelia have what are called septa. In other words, every while, as the hypha grows, you have these little divisions. In other words, one cell grows and it divides, and the next one grows. There's no cell divisions, so it's not soil fungus. It is for sure blood cells. Here you have from a nanotyrannus blood cells. You still have collagen. Okay, if this was fungus consumed collagen. There's no way you'd have collagen on it if it was fungus. And this is the best picture of all because this is a little bit of bone matrix left. And when looking at this through the microscope, I could focus through this bone. It was clear enough to see through. You could actually see the vessels going through the bone. Here's some more collagen. And so you wouldn't have collagen if you have fungus. Here's some of those little knobs that you saw in some of Schweitzer's work, and you can see in the canals. So, and this is the confirming thing. At first I wasn't really sure, and I had an idea what it might be, so I consulted an anatomist who knew more than I did. And it turns out this is what's called a vascular stop valve. Okay, why is that important? Okay, vertebrates, humans, animals, none of us have a heart strong enough to pump the blood through our bodies and back up to the lungs. Gravity is strong, and the larger the animal, the more you need these. So what happens in order for blood to circulate properly is that blood is pumped through the artery to the bottom of the foot, for example, and then as it comes back up the vein, as soon as the heart rests, it would fall back down. The heart would pump, it would try to go up, and then it would fall back down. So what happens is the veins are full of these stop valves. So as the blood is pumped up through the vein, the blood is allowed to go through this little valve, but when it tries to fall back, the valve closes. It's like a little stop valve. And you can actually see a little tissue of the valve itself in the middle. All these vessels have been liberated a whole bunch of these beautiful little stop valves, which you would only have if you were in the presence of blood vessels. Here we have a whole bunch of them. You can see where this was all the way open, and this is another osteocyte here. There's another one down here. This is very important because this is fibrous collagen, a beautiful piece of it, and you have a blood vessel going right through it. Well, if that was a fungal body or a hypha, it would consume that. Collagen. Here's a good example of a little piece of collagen. You can see how the protein on the top is balled up in these little balls. It doesn't like the osmotic pressure it's in. This is the first time this has ever been seen. I've gone through the literature, I've found no other example of this. This is probably the most amazing thing as far as rarity, probably the only time it's ever going to be found. And this is bone matrix here, this is some collagen here. Anybody have any idea what this is? Okay, we're going to put it under polarized light and see if it has birefringence. And it does. It's bright. Tendons was connected to bone. Okay, this is a little bit of... see, this thing does this to me. Tendon is connected here. Tendons are very birefringent. That means they respond really bright under polarized light. You can see the bone is not so bright. The collagen isn't as bright, but it's also birefringent. The tendons are very birefringent. So we have a tendon still connected to bone. And this is an extant modern alligator. You can see the tendon is connected. This is bone material here. Actually, the color is from the collagen in the bone. And here's more collagen over here. And this is the connection of the tendon. Here's a admonosaur. All these are tendons connected to the bone. Okay, this gets even more amazing. Something I never expected to see. Okay, imagine the bone with these canals, and here's a canal here, and then another canal. This canal is connecting to this canal. But all the bone is dissolved away. Now you only have left what was in the canal. And here you have a whole bunch of clotted blood that was in that canal, and it spilled over into this canal. Now, amazingly, a little bit of the bone matrix is left from this canal. And so you couldn't have designed it. You couldn't have asked for anything. So you can actually see the clotted blood here and how it spills into this canal over here. And these are all the erythrocytes, or red blood cells. That picture is mind-boggling. You wouldn't expect to see red blood cells, let alone a beautiful example like this. Here's a closer image of them. Okay, a little bit of background. Proteins found in previous research. Okay, when Mary Schweitzer did her work, she isolated 4 proteins. And since then, other researchers isolated more, including the top 4. Collagen, type 1, 90% of material found is collagen. Actin, which is a cytoskeletal protein. Actin is the protein that makes up the little microtubules in cells. Tubulin, another cytoskeletal protein. Histone is a DNA wrapping protein. Myosin is an actin binding motor protein associated with muscle. Tropomyosin is a cytoskeletal protein associated with the actin, and osteocalcine. Osteocalcine is a mineral that binds, is a mineral binding protein. Has mineral binding properties. Other proteins are proteoglycans, mostly found in blood serum. Lumicin is a keratin sulfate. Biglycan is another non-collagenous protein. Chondrodrian is associated with cartilage. Non-collagenous. I can't talk. I'm getting so dry. Okay, better. Okay, non-collagenous proteins include the following: albumin from blood serum, glycoproteins from blood, prothrombin, which is associated with the coagulation factor, and hemoglobin, and mostly heme. Heme is the iron pigment portion of the hemoglobin, and you'll see that all over the place. Also, some DNA fragments have been found. Most abundant protein, of course, is the collagen, type 1, alpha 1 and 2. Most collagenous protein, non-collagenous proteins, biglycans, glycoproteins, and serum albumin are from blood serum. Albumin, however, is 50 times more concentrated in bone than blood due to its binding to hydroxyapatite. Hydroxyapatite is the crystals, the mineral part of the bone. Normal collagen will completely degrade within 1 million years, and that's in perfect conditions. 4 million years if you store it in a stainless steel can in liquid of nitrogen. Nobody's ever done it. Other proteins like actin and myosin last only a fraction of that time, maybe 50,000 years under perfect conditions. Proteins are very moisture and temperature sensitive, and as well as radiation and pressure sensitive. Okay, after the initial effects of taphonomy, entropy and long-term degradation take over. Okay, this is an important slide because this is some work that Dr. Mary Schweitzer did. We're talking about proteins and the DNA. Right now we're going to look at some of the work she did in 2012. She did the work in 2012. I think she published it in 2013. Maybe it's 2012. This is the reference, so it's 2012. Anyway, Schweitzer a successful extracted short segments of ancient DNA from dinosaur cells. Okay, this is a little confusing. All top 3 are from her T-Rex. All B, E, and E are from a Brachiosaurus. This is what it looks like. And all of these down here are from a modern ostrich. Okay. All 3 cells reacted with the green glow when using ultraviolet radiation. This is an antibody to DNA showing localized binding. So you have DNA register on a T-Rex, DNA register on the Brachiosaurus, and of course a large signal from your modern ostrich. Okay, the red signal here, this is from the pydium. It's a DNA intercalating dye. It will only stain DNA. Again, you have a reaction in the T-Rex, a reaction in the Brachiosaurus, and a big reaction in the ostrich. So DNA even still exists. Now there's now clear evidence of not only intact dinosaur soft tissue and cellular structures, but subcellular structures, sequenceable proteins, and even fragments of DNA. Okay, now that was the work that I did on Mark Armitage's efforts. We both worked on that project, but he did the original work, and I ended up getting some samples and took some pictures for him. So that's phase one. Now phase two, this is search of soft tissue in ground sections of fossil bone. The sections of bone are between 50 and 100 microns thick, are studied under the polarizing microscope. This the purpose of the study is to establish if original soft tissue can be found in situation using untreated and original fossil thin sections. In other words, we're not going to dissolve them in acid. It's easy to find soft tissue when you dissolve the bone away. It all ends up on the bottom of the dish. Now we can see if we can find it in the actual bone in situation, see what it looks like. None of this has been published, and I haven't found any other work. You're seeing all this for the first time tonight. This is what typical trabecular bone looks like, or cancellous bone. Same thing. Spongy bone, you saw in the original review what spongy bone looked like, and this is what it looks like in the microscope. This is from a Edmontosaur, and this is Jurassic, by the way, so it's about 140 million years old. And this is low power. You can see you have osteon here and a canal. These are medullary cavities, these bigger ones. There's an osteon here and a canal. So you can see morphology is still there, and you can see the color. You can see all these little black dots are osteocytes still in the bone. This is an important picture. This is from our Triceratops. And you can see there's a lot of color and a lot of things to see. What is it? This is a section of compact bone from a Triceratops. The section is photographed under polarized light microscopy. The birefringence, or the bright color, is due to the orientation of collagen fibers in the bone matrix, which differs between successive layers. Remember, so I can show you here. Okay, remember the osteon? Each lamella or layer had the collagen oriented in a specific direction. Okay, you can see that in this picture due to the fact that it's taken a little bit of angle, so you're getting a little bit of one lamella here and another up here. And what's happening is collagen is a pleochroic substance. That means it has a different color depending on the direction you're looking at it with polarized light. So if you take this slide and you rotate it, the green will be green in one position and the red will be red. And when you rotate it, these colors will switch. That's pleochroism, and it will produce two different colors based on its orientation. So we absolutely know for sure that we have collagen because you have pleochroic reaction birefringence. The bone will just be light gray. You'll have some birefringence, but you won't have these beautiful colors. You can also see the fibers here, and you can see all the osteocytes are going off to the left, they're green. All the osteocytes going off to the right, they're nice and red. So we know this bone is full of collagen. And this is our Triceratops. Here we have an osteon with a Habersian canal. Here we have a canal on a different angle because this is a different layer here. Another one up here, another one up here. Okay, this picture is a mosquito larva. Why is that there? Is because you can see these are all tendons in the larva. And you can see the connective tissue, which is what collagen is. It reacts beautifully to polarized light. It's very bright. It's a very nice color. So this is an example. This shows your connective tissue is going to be very birefringent. This is another picture of a Triceratops. Here we have a canal with heavily clotted heme. This is all clotted blood in here, and osteocytes throughout the matrix. These aren't, and if you notice, these aren't hollow. These aren't little hollow lacuna with nothing in them. They have a dark colored material, oxidized protein. This is a Haversian canal. There's an osteon here with one lamina here. You can see that at this angle it's green, at this angle it's red. Okay, how do I know this is? How do I know this is collagen? Okay, this thing changes on me on its own. It has a brain of its own. Under polarized light, collagen is very birefringent, bright color. When you just use bright field, no polarizers, you don't see the collagen. So we know there's more in that bone than just bone matrix. See how the beautiful color, collagen? Here's a more trabecular bone, and you can see a canal here, and you can see in this direction it's red, this direction is green. You can see the fibers beautifully. You can see as the bone turns, you can see the color changes. It's the same collagen fiber, it's just that you're looking at it at a different angle. This is a wonderful picture because it shows beautiful collagen, but it also shows sediments. These are tiny little quartz grains. These are much smaller than the quartz grains you see on the sand and the beach. These are very microscopic. And they're the tiniest grains that get infiltrated all the way to the canals in the bone. Again, beautiful. And look at all the beautiful osteocytes still in that bone, with all the filopodia still intact. This is interesting because this shows a couple of layers. You can see this layer here is the collagen is going this direction, and this layer down here and up here, collagen is going that direction. And when the bone was ground, it was just lucky enough to have just this top piece ground off. So I can look into the bone like a little window and see all the beautiful osteocytes with their filopodia intact. So everybody's kind of getting the idea of how we determine we have collagen. Here is a really healthy, high magnification group of osteocytes, all still connected with each other, and filopodia still intact. This is Jurassic, 140 million years, if you believe that, if you trust the geological column. This is back to our Triceratops. This is only about 66 million years old. But we have something really interesting. If you notice, these lacuna still have something in them. Okay, they're not beautifully filled. These are fatty acids that have turned to concretions. It's called grave wax. They're hard, but it's still organic, and it's still got organic material in it. These are closer, and you can see all the filopodia are still in the bone. Okay, what I'm interested in is I want to study the inside walls of the endosteal surface of the canals and cavities. And the first thing, let's find out, is it the same material as the bone, or is it a different material? Okay, you can see all the beautiful osteocytes, and we'll put this under, this is right field, we'll put it under polarized light, and we'll see that the bone is one color, and this material here, which is a calcified protein, doesn't polarize like this. This is using a first order red compensator, which makes the background red. It takes you from extinction to the first order color, which is red. Which is, I'm not going to get into that, but that explains the red background. Okay, let's start our study of the walls, the endosteal walls of the cavities. You can see here flakes of endosteal proteins persist on the inside walls of the cavities. These little flakes here are protein. Some over here, some up here. It's calcified collagen. This is what the bone originally looked like. These endothelial cells are all on the endosteal surface of the canals. So we know that protein lined these canals at one time. Closer look. Okay. I have equipment, software that will analyze optical density. Okay, the densest material optically in the picture are red. The neutral or medium is green, and the least dense, almost transparent, is blue. The black is neutral. If it's 100% transparent, it will go black. If it's black, it will go black. So ignore the black. What we're looking at is the proteins have a higher density than the bone matrix. But you can still see all these beautiful little filopodia. So this, I'll refer to this as a density map. I wanted to say this is something else that's kind of rare. Almost all the canals are either all the way cut through like this or sliced at an angle. This is the only canal that went from one side of the section to the other intact. And so I want to look at the walls of that canal closer. Okay, well, look at all those osteocytes. Okay, you can see that the inside wall is lined with a fibrous collagen. And you remember, old proteins that are perturbed want to roll up in little balls. Can't say for sure what that is because I don't have Raman spectroscopy, but I can see the fibrous material that's still there. This is an osteocyte, right? I mean, erythrocyte. This is even closer. You can see the fibrous collagen lining the canal. This is a capillary canal, very tiny. This is really a tiny little canal. And I have software that will turn it into a three-dimensional topographical map, which is you can kind of map out where what the shape is. Another canal, all this beautiful collagen. Now I'm going to subject that to the density map, and you can see the areas that are dense. You can see little bits of density in the. Okay, this is a really beautiful canal with a lot of collagenous lining left. This is a closer picture. You can see through the wall here. You can see the osteocytes in the bone on the other side, but you can see that this material, this fibrous collagen, is beautiful. This is a whole bunch of fibrous collagen after the bone's been dissolved away. This is a much better condition than this is, but it gives you an example of what it looks like. But this is that was out of situation. This is in situation. This is while it's in the bone itself, undisturbed, no acids, nothing. This is almost a complete. This canal is almost completely intact. Its original endosteal protein, collagen. This is an electron microscope. This was taken by Mark Armitage. This is one of the canals that were sticking out of a bone that he processed. And this is the collagen. And this is what it looks like with an electron microscope. You can see it's connected to the wall. This is what it looks like with a bright field light microscope. That's an electron microscope. Okay, I want to study this little canal. This is a section here. You can see fibrous collagen here. What is this material on the side? We'll look at it a little closer. You can see along here it's interesting with near UV. You can see it even closer. These are perfectly preserved, possible calcified erythrocytes, but the morphology is still there beautifully. You can see the filopodia and the osteocyte over here. Another canal with a little clot of blood. Now what's interesting is you can see all the fibrous collagen left, but when blood cells are perturbed, when the osmotic pressure changes, they start forming little blisters. If you've ever looked at them, I don't know if you've looked at them with a microscope, these little blisters are still present, and that's absolutely amazing to see that they're still there. This is with the topographical software. You can see the blisters a little better, and you can see the shape, the three-dimensional shape of the cells a little better. I ignored this. I thought it was just iron oxide because I couldn't really see any morphology. So I decided one day, I said, I'm going to look at it a little closer, and I looked at it and I said, it's probably just iron oxide. It doesn't look that interesting. Then I started looking at it really close. Pick out one of these little rings and look carefully. It's all Rulio blood cells, all blood cells, all connected together. Pick out another one. Let's see, I don't know, this one up here. You can see when you look really carefully, it's a little blood cell. You look at it really close, you can see the blood cells, see the little cells everywhere. Turns out it wasn't iron oxide. It's actually calcified blood. This is my favorite. This is another a little vascular canal in the bone. And what's important about this is that to preserve this soft tissue, you can't use the standard protocol in making your sections. A regular thin section. Let me go to the next slide. Okay, a regular thin section is cut and then it's fixed. It's polished, cemented to a slide, and then it's embedded. The embedding is in some cases it turns into a hard plastic. So when you grind the section down, you can get all the way to 30 microns, which is very desirable, and it won't fall apart because you've got that plastic embedding. But then you have to remove that plastic embedding. You have to dissolve it out, and when you do, it carries all these little blood cells. It rips them all away. Okay, so you can't use that protocol. And the next thing is you decalcify. A properly decalcified bone section, you want to get rid of all this calcite that's been dissolved into the canals. This calcite dissolved in the canals. If you get rid of it all, you lose all your soft tissue then too, because this calcite acts as a cement holding all these little cells in place. If you dissolve that cement, you dissolve that calcite, a lot of this stuff, see, it will just be gone. This is about 80% decalcified. I stop at 80%. That way you can still see calcite crystals left that have been dissolved, but not all the way. Because if you dissolve them all the way, you run a chance of losing. This is, you can see all the blood cells still in this canal. But you can see they're all cemented because the calcite is still there. Look at this perfect osteocyte with this philopodium. All these cells are the right size, the right shape, they're in the right place, in the right color. This is the density map. Now the reason this is important is because blood cells are going to still have hemoglobin, which has a lot of iron oxide, which by now it's not going to be regular iron, and iron is denser than calcite. So your densest material in this image are blood cells. That's if the blood cells are still intact. If the blood cells are completely calcified or liquefied and all the hemoglobin is gone, then they'll be the same as the surrounding tissue. Okay, here's the illustration. Here's a blood cell inside this little ball of calcite. If that calcite would have been completely dissolved, that blood cell would be gone. If you look at the density map, see how dense the blood cell is? Here's a little shaving of a canal, now here and now here, and you can see the fibrous collagen in the background, and you can see how all these calcified blood cells are still stuck to the collagen. Confirming their density is much greater than the collagen with the density map. You can see through the bone to the osteocytes. See all these beautiful blood cells that are they still have they're encrusted in a coat of calcium or calcite, calcium carbonate. Here we have sediment in the fossil. This is one of my favorite, a little blood clot. It's attached to this matrix here. This is a very high magnification. This is about as high as magnification you can go with the special objectives. The advantage of the special objectives that I use means that I can take a picture to a very thick slide or without a cover slip. The disadvantage is that I can't get really super high magnification. You only can go up to about 400x. This is bright field. This is with phase contrast, which means that you added contrast to it, so you can see all the detail a little bit better. You can see in the cells themselves a little better. And then finally, near UV, you can actually see the fibrin. The fibrin is a protein that's produced by the body to acts like a net that produces a clot. And if you look really carefully, now there's no guarantee, it's just what it looks like. The morphology looks like the fibrin is still present. If you look really carefully, you can see all the little little fibrous material holding all these blood cells together. So even the fibrin is fossilized and remains. This is an outstanding classical little blood clot. The Rousselot formation of the blood cells, the fibrous collagen all around. This is even more better. This is a piece of trabecular bone. The canal starts here, runs across here, and there's a little flat spot here where the blood is settled. And if you look at it close, if you've ever looked at dried blood under the microscope, you know exactly what you're looking at. Now what I did with this, because this is so unusual and so incredibly rare, this is looking through the bottom of the slide, looking through the whole thickness of the slide. The next picture, I turn the slide over, and now you can see the canal itself. This is from the other side. You can see all of this calcified cells over here and the blood clot. And beautiful, beautiful, beautiful resolution. Okay, now we're going to go way, way back. This is from a Camarasaurus, which is Jurassic, 140 million years old. And this is another interesting series of pictures. This is a Herbertian canal here and here, and this is a Volkmann's canal connecting the two canals together. If you remember your our bone histology, this is a lamella, a concentric lamella around the canal, and this is all clotted blood, here and here. Okay, are you sure it's clotted, Jim? I said, okay, well let's look under hypothesis. Yep, there are the blood cells. And still cemented to the wall of the cavity are individual blood cells. Now, are they blood cells? Let's check the density, and yep, they're still full of iron. Now, to compare that, to give you a comparison, this is a tiny little capillary in the bone, and this is our blood cell structures that have no heme or iron pigments. These are the most lithified cells yet found by the author. Should not show the same density as the well-preserved cells. Okay, they look like blood cells, but when you put them under the density, they're not. They're calcium carbonate, just like the surrounding area. So the blood cells have been completely replaced. These are actually fossilized blood. This is near UV, and calcite, calcium carbonate, glows a little bit with near UV, so you can see it's glowing. That confirms that it's calcium carbonate. Okay, this is our perspective. We're almost done. What are the chances for original proteins to survive geological time? What does a dinosaur cell have to endure to survive and remain in good condition through eras of time? Conditions of recovery. The two worst agents of decay are heat and water. Our Triceratops bone was recovered from Montana less than 2.5 feet under the soil, with a part of the bone exposed to the atmosphere. Long summers over 100 degrees and winters are well under freezing. The freeze-thaw cycle could occur up to 50 times a year. In other words, freeze and thaw, freeze and thaw. Hell Creek is also known, recognized as a megathermal environment during the summer. So you have a tremendous amount. Why is this freeze-thaw cycle so important? Exactly. What happens, you have what's called microexfoliation. And what happens is if you have a crack and water gets into it and it freezes, it expands and widens, breaks the crack open. It thaws, the water goes out, goes away. More water comes in. If it freezes again, expands more, it makes the crack bigger. It only takes a reasonably few freeze-thaw phases to break it completely down. Break it up into a bunch of little pieces. So the fact that this bone was found at the surface meant it was exposed to the freeze-thaw cycle for, would you say 70 million years? I doubt it. Exposure to saturated moisture may occur several months of the year, so it's probably wet several months of the year. Exposure to UV radiation and environmental radiation is a big problem. The soil ecological recycling system will assault organic sources 24 hours a day. saprophytic organisms, fungi, plant roots, larvae, and boring organisms will all be able to work at that level. Now, saprophytic organisms are bacteria mostly, organisms that eat dead tissue. Chemistry and time, taphonomy, predation and decomposition. This is an example of taphonomy when we talk about it. This is only a couple of weeks laid out on the ground. Doesn't last very long. Diagenesis, we talked about that a little bit, describes the changes and alterations that take place on skeletal remains. This is a cumulative physical, chemical, and biological environment. These processes will modify a bone's original chemical and structural properties and will govern its ultimate fate. The longer the time period, the more alterations you should encounter. Skeletal diagenesis is the first step in the process of involving the elimination of the organic collagens by the bacterial collagenases. These collagenases break down protein into peptides. For bone to turn to stone, all the collagen needs to be gone. Autolysis, self-digestion through the action of its own enzymes causing cell destruction. This occurs in dying or injured cells. It's initiated by the cell's lysosomes releasing digestive enzymes into the cytoplasm. These enzymes are released due to the cessation of active processes in the cell. It helps the immune system break down and liquefy dead tissue. It's a self-limited function, but it really gets the decay process going. Proteolysis is what happens to proteins, usually saprophytic, anaerobic, and aerobic bacteria. Putrefy. Hydrolysis, biological hydrolysis is the cleavage of biomolecules where a water molecule is consumed to affect the separation of a large molecule into its component parts. This process will continue indefinitely and is initiated from the beginning with the exposure to moisture. Adipocere, we talked about that a little bit. Grave wax, it's a waxy-like organic substance formed by anaerobic bacterial hydrolysis of fatty acids in corpses. It is in its formation, putrefaction is replaced by a permanent, firm cast of fatty tissue. It forms a water-insoluble, waxy concretion with a tan color. You saw some examples of that. Oxidation, everybody knows what that is. It happens all the time. It's very destructive. We talked about the freeze-thaw cycle, micro-exfoliation. Leaching, this is when the groundwater is, if it's on the acid side, it will leach all the minerals and biochemicals out of the fossil. This is very important, natural radiation. Environmental radiation will relentlessly degrade bone tissue, especially in bone. Bone absorbs uranium and thorium. Over time, this internal dose will wipe out all biomolecules. Dinosaur fossils soak up radiation so much so that in many cases they will react to a Geiger counter. And in some cases, the bone has been painted in lead to protect the public from exposure. Temperature, high temperature accelerates putrefaction and rotting tremendously. What about that? Chemistry. Pressure. Okay, this is another important one. After deposition, sediments are compacted as they are buried beneath successive layers and cemented by minerals that precipitate in solution. In this situation, pressure melt. That's what's happening. Pressure melt can occur in which individual grains who are under the greatest pressure can dissolve easily into the groundwater. The dissolved mineral then precipitates into the pores of the bone, which causes diagenesis. Pressure is not good. Rapid burial is essential for fossilization. You guys probably know all about that. Confirmation. Okay. I had a huge list of references of other work in this area where soft tissue has been identified in peer-reviewed literature. So there's a lot more now, but by May 2018, 26 scientific journals, including Nature, had confirmed the discovery of biomaterial in fossils. This includes many different genus of dinosaur. A list of over 100 journal papers announce these discoveries and support the increasing common appearance of soft tissues. Okay, this can now include our Triceratops and dinosaur, which date to as old as 80 million years. Now, this is one of the fun things, geology and plate tectonics. Okay, we had represented in the presentation the Admonosaur and the Camarasaur. These are Jurassic. This is what the world looked like if you believe plate tectonics. The Jurassic world, North America, Africa and South America were one continent, and completely different world than now, obviously. The Cretaceous is where our Triceratops came from, and our Nanotrice. This is what it looked like then. Still a completely different world. You know, Africa and South America are only a few hundred miles apart. Europe, hardly an inland ocean. And North America was made of this big island here and these three islands. Okay, where was our Triceratops found? It was found in Montana, and that's approximately where Montana is. So keep your eye on that T, and let's look at the world today. Oh, our Triceratops never lived in Montana, lived in Nova Scotia. The continent has moved 3,000 miles off to the west, carrying our Triceratops with it. Okay. Okay, our Triceratops was transported to a grinding recycling process for nearly 3,000 miles. He would have endured over 1 million continent-moving earthquakes with shifting stress, fractures, and pressure variations. How many times would have been turned over in his grave? This would not even take into account the millions of years he would have been buried under countless tons of inland ocean waters. Well, this is what they tell us. Okay, another dilemma. This is taken at the Museum of the Rockies in Bozeman, Montana. And the Rocky Mountains are in the background, and that's the subject of this paragraph. Rocky Mountains are on their second generation since the Cretaceous. The Lamerid erosion was a period of mountain building in North America which started in the late Cretaceous 70 million years ago and ended 35 million years ago. After this mountain building, the Rockies were like Tibet, a high plateau, probably about 20,000 feet above sea level. In the last 60 million years, erosion stripped away the high Rockies, revealing the ancestral rocks beneath, and forming the current landscape of the Rockies. Millennia of severe erosion in the Wyoming Basin transformed intermountain basins into relatively flat terrain. Now, our Triceratops was found in the northern Wyoming Basin. Over half of the Rockies have eroded and deposited massive amounts of sediments across the surrounding. In fact, most of the Great Plains that are all east of the Rockies are erosion products of the Rockies. So the question this brings up is, if our Triceratops was buried during the Cretaceous, before even the mountains were born, before they were even built up, and then the mountains rose and then eroded away on top of the Wyoming Basin, according to geologists, our Triceratops should be under about 1,500 feet of sediments. But it wasn't. It was in a floodplain. Notice I said floodplain, right on the surface. So, was it buried after the mountain building or before the mountain building? So, the question is, there's a lot of conflicts, and I'm pretty sure that our Triceratops actually died during a flood. I think most of you agree with that. And probably wasn't 70 million years ago. So we still have soft tissue left, which shouldn't be there if it's that old. And we have conflicts. Discovery of dinosaur soft tissue is not yet adequately explained. The science of tissue decay, which 150 years of experience behind that, does not permit these long survival times. Even resistant proteins should be limited to the rarest of discoveries. The frequency of recent soft tissue discovery now points to the possibility that it's more common than ever thought possible. And that is it. Time for questions. To leave tonight, I've got over here an exhibit set up. I don't think we need this on. Okay, I've got some dinosaur bone. I'll bring up on this little monitor. I've got a stereo microscope with some rib material from the Triceratops. And I've got one of the dinosaur slides on the far end. Just don't pick it up because it's extremely expensive. But it's there to see. You can sit down and look at the fossil under the stereo microscope, and I will start that machine, that microscope. It's a polarizer microscope, and I'll put one of the blood clots you saw on the thing up on the screen. So I'll take 1 or 2 questions, then we'll take a break, and then if anybody has more questions, we'll take more later. Any questions?
Where does carbon-14 fit into this?
Well, carbon-14 is a dating method that measures the decay of carbon-14 over a period of time. And in the literature, there's been, I think, 32 dinosaur fossils that have been anonymously submitted for carbon dating. And the youngest is 18,000 years, 16,000 years, and the oldest is about 42,000 years. So all of the bones that have been carbon dated so far have not come anywhere close to being millions of years old. You know? And the other thing is, paleontologists ignore this completely. They think that that it's contamination. However, for some reason, which is very confusing to me, is that anthropologists totally and completely accept the carbon dating from ancient bones that they get. So their dates are fine, and some of their bone material that I looked at is in worse condition than our dinosaur bone is. You know, so one group of scientists accepts bone dating, fine, and paleontologists won't even do it. So who knows?
So has any of the soft tissue been carbon dated or no?
Well, soft tissue would generate great carbon dates. The soft tissue is where the carbon would be. You know, the hydroxyapatite, which is a mineral, that's not going to have any carbon in it. You know, the carbon-14 is a process that remains in tissue. So carbon-14 would tell you that that bone still has tissue, or it still has the carbon from the tissue. In other words, it's not, you know, the carbon-14 is good for about 50,000 years. I mean, it can be done a little later, but it's considered accurate up to about 50,000 years. And so paleontologists say there's no reason to do carbon dating because dinosaurs are millions of years old, so the results are not, you know. So that's the thing.
Well, what in your opinion would be a more accurate date range? Take your Triceratops. How old do you think he was?
How old do I think this? The honest answer is I have not the slightest idea.
I mean, what would, even a ballpark? I mean, thousands, millions?
The amount of collagen left, you know, under the circumstances, I didn't read it, but under the circumstances, collagen has a half-life of about 2,000 years under the circumstances you would expect. In other words, if you're out in a field, you die and you get buried with mud in a flood, and you're not isolated from taphonomy really, then the collagen should last a half-life of about 2,000 years. So collagen should be completely gone under those conditions, probably within 10,000 years. Now, the conditions that I said up there, a million years, that's perfect condition. That's in a stainless steel jar in liquid nitrogen, you know, or in a freezer. Now, even in a freezer, if you take a steak and you put it in your freezer and you say, "Oh, I forgot it. It's 2 years old," it's going to be considerably degraded, even though it's frozen. You know, so no, probably 8 to 10,000 years, based on just the science. You know, now I believe in the flood. So I would, if I had to say, I would say that all these dinosaurs, because they're all found in floodplain environments, I would say that it died in the flood. So how old is Noah?
Could I get an add on?
Yeah.
In your opening title page, you had an archaeological, it looked like, you know, the Stegosaurus?
Yeah.
that was an archaeological ruin, or?
Yes, that's from a temple in Cambodia. The temple was built 850 years ago, and it was built in a jungle environment. There are no fossils ever found in a jungle environment. It's too moist. So that there's you can't say these people would have found a fossil. You know, so for them to have actually carved a Stegosaurus is unique. It's one of the only living things that looks kind of like a rhinoceros, maybe, but it has these big solar plates all on its back. You know, you can't really mistake that for anything else. You know, there's nothing else that has it. You know, so you're pretty safe in saying it's a Stegosaurus. So how did these people 850 years ago know that? How were they able to image a Stegosaurus and then revere it enough to carve it onto their temple? It's a column at a temple in Cambodia.
Yeah, so the tendon that was found on the bone, that's connected on the outside of the bone, right?
Yes.
So it's not encased in the bone, it's not protected, and yet it's still there.
Yeah, that's right. It's still there. Now, the nice thing is that tendons are not affected by the acids, by the ETA. It's not going to dissolve away. The bone dissolved away, so there's just a tiny bit of bone left. You know, but yeah, if it's there, you should expect to find it. You know, but no one it's the only time no one I know of has ever found tendon.
But it's still affected by the elements, and
it's still affected by diagenesis and taphonomy and decay and you know, yeah, it's still very much affected. It should be gone.
Right,
You know.
because I'm thinking because it's outside the bone, it's got a better chance of disappearing.
Yeah, it has no chance of. You would think it would have no chance of being here. You know, so yes, but it's what it is. You know, and that's why I wanted to get the pictures close so you could see them, because the proof is in the pictures. You know, if I told you I found tendons of dinosaurs, everyone would laugh at me. Oh, now here's the picture. Here it is. Here's the test. You know, you can look at it yourself. You know, so.
Since you have the soft tissue over there, does that mean the genes?
No.
I believe we have 23.
No. There's going to be. Dr. Schweitzer found fragments of DNA. Okay, a gene is a whole section. It's a whole section of a chromosome, and a chromosome is a DNA molecule wrapped up in a little ball like this, and it makes up. And you know, she's found little fragments of DNA. There's no gene left, and there's no chromosome left, or anything like that. It's just fragments of DNA. So no, there's no chance of sequencing. There's no chance of like. Jurassic Park. You know, so yeah.
When you dissolve the bones, how do you stop at 80%?
You know, well, what you have to do is you have to monitor it. You have to put it in a petri dish with the lid on it like this, and you put it under the stereoscope, and you keep your eye on it. You know,
Egg timer.
Yeah. Well, the problem is every bone section has a different density. I mean, in other words, I can't say this is the equation. It's going to be 20 minutes every time. No, 20 minutes may completely dissolve one and hardly even touch another. You know, because each the minerals that are precipitated into the bone, you know, generally speaking, most of it is calcium carbonate. But all these minerals have other things in them. For example, siderite is calcium carbonate with iron in it. You know, so that's found in fossil bones. You know, you're all going to have different compounds that are going to change the timing equation. So you can only really do it correctly. And when I say 80%, it's between 70 and 85. You know, you can't just sit there the whole time. But you know, so you have to kind of work at it.
Quick one. In reading about this kind of work, it seems like the people who are still saying you've got billions of years on these dinosaurs say we really have to take a look at how things decompose better.
Yeah.
Is there any serious work going on in that area?
Oh, they've come up with, Mary Schweitzer came up with her iron chelation theory, which is it's a laboratory experiment where that is completely unrelatable to actual fossils. What she did was she took let me make sure I get this correctly. She took ostrich blood, okay, and the first thing you have to do is you have to stop blood from you have to put an anticoagulant in, which is never going to happen in nature. Otherwise, the blood will coagulate and bind together, and you can't get the hemoglobin out of it. You can't get the iron out of it. It locks up, as soon as it coagulates. So she had to take the blood and immediately put anticoagulant. And then she had to next thing she had to put it in a centrifuge and separate all the other stuff out. You can't have the proteins, you can't have the albumin, you can't have the white blood cells. All that stuff had to be taken out. So she had nothing but the red blood cells left, because all that other stuff would mess it up. Then she took the red blood cells and lysed them and separated all the membranes away. So she had nothing left but the iron. And then she took the iron, the hemoglobin, and she put and then she put blood vessels, fresh blood in that one, and then she took another one with just plain water blood vessels in, and the one in water decayed after after 2 years, and this one is in better condition. Well, yes, it's in better condition because it's not in water; it's in iron. It's in iron fluid, and the process is going to be different. But none of those conditions can possibly exist. You can't stop a dinosaur that dies from his blood. You can't stop his blood from not clotting. So she's saying it's iron chelation. In other words, it's protein. The protein is bound together. It's a kind of a fixation, like formaldehyde. But it's impossible. It's just a lab experiment. To come up with a reason. There's another one now. It's called polymerization, where the material somehow gets heated up to 200 some degrees or whatever, and then the proteins chelate, and now they can last a lot longer. Well, none of those conditions ever happen. Well, you can't heat the protein up to chelate it. I mean, not to chelate it, to turn it into a polymer. From a monomer. In other words, two monomers turn into a polymer. It's kind of like, it's an analogy. What they're trying to say is that, use an example of an egg. If you take an egg and you crack it and you spill it out, the egg white is really clear. You drop it on the soil and it soaks into the soil, and all the bugs get to it, and it doesn't last very long. Well, they say, well, by polymerizing it, it's like taking the egg and boiling it. And now when you crack it, it's a nice hard. Now it's actually got a real egg. But you drop that on the ground and it will bounce and maybe break in half. And so now it's going to last. Well, it's not, because nature doesn't work that way. The hard-boiled egg in real nature will probably last less time than the other, because a raccoon will come along and eat the whole egg. Either that, but on a microscopic level, there's a bacteria that eats everything. So if you have a protein that's not polymerized, you have a certain type of saprophytic organism. But then you have iron-eating bacteria. You have sulfur-eating bacteria. And so now you have this polymerized. Now nature just has something else to take care of. And so the only thing that will do is it will help you on the short end. In other words, in the short run, it will last a little longer, but in the long run, it will be worse. It's the same thing with the iron chelation theory. It's good for the short term, but it's like iron chelation is like rusting. Rust, after a while, it completely falls apart. It lasts longer in the short term, but in the long term, you gain nothing. So there really is no explanation yet. There's no real good explanation of how these things. There are dozens of people frantically trying to find a solid how these things survive for millions of years, because they have to keep their millions of years. That's the paradigm. And, every article written is written around these millions of years, around evolution. Every article you publish is this is another explanation of how this animal evolved. So you take that away and you go back to Newton's time. So anyway, I'm going to turn this stuff on. So if anybody wants to see the exhibit, let me get this going, and I'll turn it back over to Jim. And if you have any more remarks, Jim. Okay, let me get this going, and then you guys can come up and take a look at the exhibit. Okay, you can see the osteocytes now. See the osteocytes in the bone? And then when you focus in, you can see the calcified blood cells. Okay. Any more questions? Well, thank you, everybody. Thank you for your attention, and thank you for letting me speak.